Dracaena. So this is the plant here, and let's move towards its section, the section of the stem. Here the transverse section of the stem is shown, and you can see that it has the young Today we are going to discuss about the important topic, that is of the secondary anomalous growth in the plant Dracaena. So let us see the transverse section of the stem here. The young stem of Dracaena is a typical monocot stem. It is a typical monocot stem, and normally the vascular bundles of monocot stem, as we know, they are of conjoint, collateral and closed type. Conjoint here refers to the situation where the xylem and phloem are on the same radius. And in case of collateral, it means that the phloem is present towards the periphery while the xylem is present at the towards inside. And in the case of closed type, it means that it lacks cambium, which is a typical feature of the monocot stem. And thus, as there is the absence of cambium, it lacks the secondary growth in thickness and vascular system, be completely composed of primary tissue. Here the vascular bundle remains irregularly scattered in the ground tissue. You can see that these primary vascular bundles are just irregularly scattered and forming this as a attacked stele. Here some monocot belonging to the family, as it belongs to the family Liliaceae, along with Dracaena, the different kind of that is yucca, agave and aloe and other exhibit a special type of secondary increase in thickness. Now let's discuss in detail about the transverse section of Dracaena stem, which is, you can see it is almost circular in outline, and next is the part that is of peridome, and in case of peridome it is composed in the case of epidermis. Actually the epidermis in the normal stem is converted in the old stem as the term peridome. So we are giving the explanation of the term peridome, which is composed of the parts, that is first of all cork, which is also known as phloem. Second is the cork cambium, which is also known as the phloem, and third is the secondary cortex, also known as the phylloderm. Here the phloem is actually a rectangular in structure, and it is thickly suberized, and obviously as it is cork it is dead, and the phloem is 1 to 2 layered thin wall, and that is tangentially elongated, and these three structures forms the peridome in case of the old stem that replaces the epidermis with the peridome composed of phloem, phloem and phylloderm. And now let's discuss about the second point, that is the cortex. In case of the stem of Dracaena, it has undifferentiated region composed of the parenchyma cells that contains the starch grains. You can see here there are starch grains present, and likewise next one, let's move towards, that is the meristematic tissue. In case of the stem, here the meristematic tissue actually is the cortical cells that are outside the vascular bundle, that are outside the vascular bundles. You can see here they are outside vascular bundles, and they form several layers of meristematic zones, and the cells, these zones are made up of actually rectangular. They are rectangular in shape and they are generally radial, and which acts as cambium, and this acts as the cambium which helps for the secondary growth, secondary thickening in case of the old stem. And let's move to the fourth part, that is the vascular tissue system. Here you can see that the primary vascular bundle here has outer, also there is the outer closing with the sclerenchymatous patches covering are seen in the primary vascular bundle here, which are freely scattered in the ground tissue. These are the primary vascular bundles which are freely scattered in the ground tissue, and they are large in size. You can see that these secondary bundles are quite large in size. They are large in size and they are present, which are large in size and typically the same, that is conjoint, collateral and close type. Conjoint is the case where both xylem and phloem are on the same radius, and collateral is the case where the phloems are at the periphery, and in case of close type it lacks cambium. In case of primary vascular bundles they lack cambium, so it is close type. And in case of the secondary vascular bundle here, you can see they are present near the periphery, which are smaller in size and they remain embedded in the thin wall tissues. They remain embedded in the thin wall tissue, and the secondary vascular bundles are actually concentric and amphivessel type. And the concentric and amphivessel type is, it means that it has like this structures, that is with the secondary phloem at the middle place, which is covered with the outside by the secondary xylem in the same form. Secondary xylem at the periphery, and at the center there is secondary phloem, and this kind of structure is called the concentric and amphivessel type, which is present in the secondary vascular bundle. These are the structures which are near the periphery and smaller in size as well compared to the primary vascular bundle. And the secondary vascular bundles here, and it likewise the another term here is the secondary phloem, and the secondary phloem is composed of short sheath tubes. They are composed of short sheath tubes elements, while the secondary xylem here is composed of tracheids and xylem parenchyma. So this is the case here. So this is all about the vascular tissue system. Now let's move to the another one, that is the ground tissue. Here the tissue extends from the inner side of the meristematic zone. This is the meristematic zone, and it is filled up with the major part. This fills the major part of the stem, and the cells of the young tissue are actually thin walled parenchymatous, and they are with intercellular space. Parenchymatous with intercellular space, and it fills up the major part of stem. And now let's move towards the secondary growth in Dracaena. At a very late stage of development, a wide zone of secondary meristem. Here the wide zone of the secondary meristem, that is the cambium here, that is present, that acts as the cambium in case of the old stem. It develops outside the vascular bundles in the parenchymatous region, and this meristematic zone cuts off the vascular tissues on its inner side, and the vascular bundles are arranged in the concentric manners. They are arranged in the concentric manners, and they are the amphivasal in nature, that is secondary phloem at the center, which is surrounded by the secondary xylem at the periphery, and the amount of the parenchymatous ground tissue also increases, and therefore that the diameter of the stem also increases. So this is the case of the secondary growth in Dracaena. This was for today. If you like this video, then just like, share with your friends. Thank you for watching.